Hello friends, I once again welcome you to Dr. Great. So in the previous videos we saw about the glycolysis part 1 and glycolysis part 2, in which we discussed the whole aspects of the glycolysis, the aerobic and anaerobic both. So in this video we are going to see about the Krebs cycle. And one more thing, if you haven't watched these two videos, that is the glycolysis part 1 and part 2, then the link is in the description below, or else you can click the I button that is above. So let's go to the Krebs cycle without wasting any time. So the first thing is that it has three names. So one is the citric acid cycle, then the TCA cycle, which is also called as a tricarboxylic acid cycle, and the third one is the Krebs cycle. And the second point is that the final oxidative pathway for carbohydrates, fats and amino acids. So this Krebs cycle forms the final oxidative pathway for the carbohydrates, fats and amino acids. So what do you mean by this point? So the Krebs cycle forms as an intermediate for the formation of the carbohydrates, amino acids and fats. The examples could be the gluconeogenesis, where the glucose is formed from the non-carbohydrate sources, then the glycolysis, glycogenolysis. And we will be studying about these concepts in the later upcoming videos, but till that time just keep in mind that it forms a final oxidative pathway for carbohydrates, fats and amino acids, like the Krebs cycle. Like the precursors have to pass through the Krebs cycle in order to form the final product, that is, they can be carbohydrates, fats or amino acids. Now the third point is the acetyl-CoA, when it gets oxidized, it forms carbon dioxide, and along with carbon dioxide it forms water. It gives us carbon dioxide and water. So why this is important? It is because the acetyl-CoA gives rise to many products such as the lipids, the fatty acids, etc. and etc. So it's helpful to keep in mind that when acetyl-CoA is oxidized, it will give rise to carbon dioxide and water. And the next point is 65 to 70% of ATP is synthesized in Krebs cycle. So the Krebs cycle is the most energy generating cycle, as it gives out the most number of ATPs, or a greater amount of ATPs, that is 65 to 70%. So the next point is it acts as an intermediate for other metabolic pathways. Like I said before, these both points are similar. The only thing is that in this point they have mentioned that this whole cycle acts as a pathway, a metabolic pathway for other metabolic pathways. So like this Krebs cycle is a center, or it's an important pathway for the formation of the carbohydrates, fats and amino acids. So the location or the site where the Krebs cycle occurs is the mitochondrial matrix. So the Krebs cycle occurs in the mitochondrial matrix, and the ATP synthesis that occurs in the Krebs cycle is by the oxidative phosphorylation reactions that occurs in the mitochondrial matrix. So site is the mitochondrial matrix, and the ATP synthesized is by the oxidative phosphorylation. And one more thing, the acetyl-CoA, that is a 2-carbon compound, the acetyl-CoA, along with the oxaloacetic acid, that is a 4-carbon compound, gives rise to the citric acid, that is a 6-carbon compound. And here the oxaloacetic acid gives a catalytic role. It plays a catalytic role, more clearly to say, like it catalyzes these reactions and the Krebs cycle in order for the Krebs cycle to run smoothly. So at last the acetyl-CoA is a 2-carbon, along with the oxaloacetic acid is a 4-carbon, forms the citric acid, that is a 6-carbon compound, and the oxaloacetic acid plays a catalytic role in this reaction. But before moving on further, please subscribe to the channel and press the bell icon to receive the latest updates. So coming to the initial step of the Krebs cycle. So in the previous video we saw about the glycolysis. So the end product of glycolysis is the pyruvate. So the pyruvate gets converted to acetyl-CoA. But between this conversion from pyruvate to acetyl-CoA, there are main factors involved in this, and they are the lipoic acid, the coenzyme A is added here, then magnesium is also helpful, then the TPP, that is a coenzyme, thiamine pyrophosphate, then the NADH is reduced to NADH2. So these 5 factors, that is the lipoic acid, the addition of CoA, the thiamine pyrophosphate, the magnesium 2+ or magnesium simply, and the reduction of NAD to NADH are the 5 main factors responsible for the formation of acetyl-CoA from the pyruvate. And during this reaction, there are two things released. One is the carbon dioxide and the other is water. So when there is a conversion of pyruvate or acetyl-CoA, so this step, that is the conversion of pyruvate to acetyl-CoA, is also called as oxidative decarboxylation. So the pyruvate conversion to acetyl-CoA is called as oxidative decarboxylation, and in this oxidative decarboxylation we get the carbon dioxide and the water released, and there are 5 factors important, and they are lipoic acid, coenzyme A, magnesium, thymine pyrophosphate, and the reduction of NAD to NADH2. So we all know that Krebs cycle is a complicated cycle because there are so many intermediate compounds and molecules, and that is why we introduce mnemonics. So as I introduced mnemonics for the aerobic glycolysis, so here are the mnemonics for the Krebs cycle too. So the mnemonic says that our city is openly kept safe, so fun is maintained or fun maintained. So what does this mean? Like, this means that the city is kept safe, and each and every city that is the surrounding this city knows it, that it is being kept safe. Like, generally it's safe, and that is the reason why the city people or city population is very happy and they are maintaining their fun. So finally it is like our city is openly kept safe, so fun maintained. So the compounds or the molecules assigned to this are, like, for our it is oxaloacetic acid, for city it is citric acid, for us it is isocitric acid, for openly it is oxalosuccinic acid, kept alpha ketoglutarate acid, safe succinyl-CoA, so succinic acid, fun fumaric acid, maintained malic acid. So our city is openly kept safe, so fun maintained. Therefore oxalic acid, citric acid, isocitric acid, oxalosuccinic acid, alpha ketoglutarate acid, succinyl-CoA, succinic acid, fumaric acid, and malic acid. So these are the main substrates or intermediates of this Krebs cycle. And as in the previous video I said that the enzyme thing, like how to derive the enzymes. So in this Krebs cycle it is very easy, and for that also there is a mnemonic. So the mnemonic for the enzymes is as follows. So after decades, darkness surrounds devils, falling death. So what does this mean? Like, after decades, the devils are surrounded by darkness and they die. So the main thing that we have to remember here is all the S here stands for synthase, and the D here stands for decarboxylase. So S for synthase and D for decarboxylase. So like it contains S, like the first letter is S. So synthase, D, decarboxylase, D, decarboxylase, synthase, D, decarboxylase, D, decarboxylase. But here there are two words that have A and F letter as their starting. So what about them? So the after stands for aconitase and the following stands for fumarase. So these two enzymes have to be remembered because they don't have any trick like synthase or decarboxylase, but yes, after for aconitase and following for fumarase. And also the D means dehydrogenase enzyme also. So the probability for the D is two things, that is the decarboxylation or the dehydrogenase enzyme. So whether it is a decarboxylase enzyme or a dehydrogenase enzyme, it will depend on the reaction. Like if the carbon dioxide is released in the reaction, then the decarboxylase will come, and if the NAD or FAD or any other substrate is reduced to its lower substrate, then the dehydrogenase will come. So the first step is the oxaloacetic acid along with the acetyl-CoA forms the citric acid. And here the enzyme that is coming in use is citrate synthase, and it is also called as the condensing enzyme. So during this process or during this step, we say that the H2O is also inserted here. So the acetyl-CoA along with the water and the oxaloacetic acid forms the citric acid, and here the CoA is released, and the enzyme here used is citrate synthase. And one more thing to notice is this acetyl-CoA is of 2 carbon and this oxaloacetic acid is of 4 carbon, and they combine together to form a citric acid which is of 6 carbon. So 4 plus 2 equal to 6. So 6 carbon citric acid is formed. So this is the first or initial step. particularly of the Krebs cycle. So as the citric acid is formed, here we see that the 6-carbon citric acid gets converted to cis-aconitic acid in the presence of the enzyme aconitase. Remember I said you the mnemonic after, so for that the aconitase, and during this step the water is removed or released, and again when the cis-aconitic acid is converted to the isocitric acid, the water again is added here, and again the enzyme that is used here is aconitase. So actually this is an intermediate step where the citric acid gets converted to isocitric acid in the presence of the enzyme aconitase, and generally there is no loss of water because it is maintained here. So the citric acid gets converted to cis-aconitic acid, it forms an intermediate actually, then it forms into isocitric acid in the presence of enzyme aconitase. The water is compensated here because the water is released also and added also. So now the 6-carbon isocitric acid is formed. So after this it gets converted to oxalosuccinic acid in the presence of the enzyme isocitric acid dehydrogenase. I said you in the previous video that the name for the enzyme is derived from the previous molecule or compound, like isocitric acid. So isocitric acid, and from the trick or the mnemonic you can identify it has dehydrogenase. So overall it is isocitric dehydrogenase which converts the isocitric acid into oxalosuccinic acid, and here the NAD plus is reduced to NADH2, and again the oxalosuccinic acid here gets converted to alpha ketoglutarate acid in the presence of the enzyme oxalosuccinic decarboxylase, and also there is a release of carbon dioxide which ultimately gives rise to alpha ketoglutarate acid. And also as the NAD is reduced to NADH2, therefore the name dehydrogenase is coming, and as the carbon dioxide is released, therefore the name decarboxylase is coming. So this can be the minute things that can be used to remember these enzymes. So finally the isocitric acid gets converted to oxalosuccinic acid in the presence of the enzyme isocitric acid dehydrogenase, and here the NAD is reduced to NADH2, and the oxalosuccinic acid is converted to alpha ketoglutarate acid in the presence of enzyme oxalosuccinic decarboxylase, and here the carbon dioxide is released. And one more thing, as the carbon dioxide is released here, the carbon gets reduced to 5 carbons. So the 6-carbon oxalosuccinic acid gets reduced to 5-carbon alpha ketoglutarate acid as there is a release of carbon dioxide here. So the next step is the alpha ketoglutarate acid converted to succinyl-CoA. So the alpha ketoglutarate acid is a 5-carbon compound or acid, and the succinyl-CoA is 4-carbon compound, and this occurs in the presence of the enzyme ketoglutarate dehydrogenase, like dehydrogenase because the carbon dioxide is released here, and ketoglutarate because the alpha ketoglutarate acid is involved here. And during this step the NAD plus is reduced to NADH2, so the NAD plus gets the energy as it is reduced to the NADH2, and the coenzyme A is released. So finally if we see, we can see that the alpha ketoglutarate acid gets converted to succinyl-CoA. So alpha ketoglutarate acid 5 carbon, so release of 1 carbon in the form of carbon dioxide, so 4-carbon compound is the succinyl-CoA, and the coenzyme is again inserted here, and the NAD is reduced to NADH in the presence of the enzyme ketoglutarate dehydrogenase. So in this process or in this step we see that the succinyl-CoA, which is a 4-carbon compound, gets converted to the succinic acid, which is also a 4-carbon compound, in the presence of the enzyme succinic thiokinase. So succinic thiokinase, the succinate dehydrogenase. So basically the succinic thiokinase is also called as succinic dehydrogenase. So there is nothing to confuse among this. We can remember the name as the succinate dehydrogenase also, or only we can remember the dehydrogenase enzyme. And here we see that the water is added and the coenzyme A is released, and here the ATP and the GDP is converted to ADP plus GTP. So eventually here we see that the ATP is consumed here, but it's not like that. We get a ATP here. The reason is this gets recycled. To be more clear, we see here that the ATP plus GTP is not consumed only, it's also formed here. So we consider this as the formation of ATP. So here ATP is formed. So finally, the acetyl-CoA, which is a 4 carbon, gets converted to succinic acid in the presence of the enzyme succinic thiokinase, or mostly called as the succinate dehydrogenase. And here the coenzyme A is released, the water is added here, and there is a formation of ATP. So here we see that the succinic acid, which is a 4 carbon acid, gets converted to fumaric acid in the presence of the enzyme succinic dehydrogenase, as the FAD is converted to FADH2, and this fumaric acid that is produced gets converted to malic acid in the presence of the enzyme fumarase, and a molecule of water is added here. Then finally the malic acid gets converted to the oxaloacetic acid, that we saw it in the starting, in the presence of the enzyme malic acid dehydrogenase, because malic acid and dehydrogenase, and here the NAD is reduced to NADH2. So finally, if we say that the succinic acid is converted to fumaric acid in the presence of the enzyme succinic dehydrogenase, and here the FAD is reduced to FADH2, and then this fumaric acid gets converted to the malic acid in the presence of the enzyme fumarase, and here the water is added here. Then this malic acid gets converted to oxaloacetic acid in the presence of the enzyme malic acid dehydrogenase, and here the NAD is reduced to NADH2. So this is the whole Krebs cycle, which I divided into parts and explained it to you. So the overall cycle is here, and with this we complete this Krebs cycle. Thanks for watching this video, guys. Like the video, subscribe to the Dr. Great channel, and show your support to me, and share this video to everyone as much as possible. And if you have any suggestions, please put that down in the comment sections below. And once again, thank you all.